A lot of times in this class, but this is, I find this really interesting. A lot of times what I see is that when we talk about a big topic, everybody's like, yeah, okay, I get it. And then when we start diving in and talking about specifics, sometimes people, including me, sort of lose sight of the big topic. So I want to remind you that what we're looking at right now are cells, including the structures and organelles inside cells. And over the course of the next several days, we're going to be looking at a few things specifically. We're going to get even smaller. We're going to look at a couple different organelles specifically and structures. The cell membrane is a structure we're going to look at. We're going to look at chloroplasts as well, and probably mitochondria. We're going to look at something called cell transport, which is how things move from one side of a cell membrane to the other, which is really critical. And as we talk about those things, though they're in your consciousness, they're going to sort of expand to be like, that's what we're doing. What are we doing right now? Cell membranes. But just remember, cell membranes is just a smaller part of looking at and thinking about cells overall. In this class, I have been relatively committed to not just having you all think about like what things are or how they work, but I would also like you to be able to do some stuff. So there is some information here that I want you to be thinking about. Cell membranes, how they work, what pieces go into a cell membrane, and how all those pieces work together to allow the cell membrane to do its job. But I also want you to be able to do some broader science. And so one of the things that we're going to look at today is how can you design an experiment? If you want to know something, how can you design an experiment to figure it out? And when you ask a question and you design an experiment, what you get directly from that experiment is not the answer. What you get is data, numbers, measurements, observations. And interpreting that data is what gives you the answer. Okay? If I want to know who's stronger, me or Ava, then I might challenge Ava to a series of arm wrestling competitions. And if we arm wrestle like 37 times, and I win 12, and Ava wins 13, and 2 are ties, then that isn't the answer to the question, who is stronger. That's data. If we want to know the answer to the question, we have to interpret the data. And interpreting the data means doing things like, well, let's see. The ties are kind of like equal, and then she won 13, I won 12, so she's like, how much? I mean, a little bit stronger, but not dramatically stronger than me. And we're going to be working on that as well today. So you're going to do some science. When it comes to the structure and function and components of cell membranes, largely this is an exercise in visualization and vocabulary. Luckily, none of it is super challenging. So here are some things to know about cell membranes. And remember, if I underline it, you should write it down. So the first thing is cell membranes surround cells. And that sounds obvious, except that in the pictures that we see, typically what we're seeing is a cross-section. We'll often look at cell membranes like this, as if there's been a cell that's sliced in half and we're just looking at one layer of it. This is a 3D cross-section. It still shows a cell that's been sort of cut in half. But this is more like what a cell membrane would look like, right? Like it's all around three-dimensionally the inside of the cell. Nothing can get in or out without going through the cell membrane. And just as a quick aside, I posed a question to you a couple weeks ago when we were first looking at microscopes, and the question was, did anybody notice that when you're looking at those cells, that if you focus on one thing, then sometimes other things go out of focus. And then when you go back to focus on the first thing, then the second thing goes out of focus. Did anybody notice that? The reason for that, which some of you know, but for me, it's always something that I forget, is because biology isn't flat. Things aren't all on one layer. When you focus up and down to see different things in a slide, you are looking at layers. That slide looks really flat. To us, it's like just a single flat layer. But if we look at it from a biological perspective, it's got a lot of thickness to it. So if you want to look at the things that are closer to the top of the slide, you focus up. If you want to look at the things that are closer to the bottom, you focus down. And this is a good illustration of what I'm talking about. If we had a cell in a microscope and we could magically look at any layer, we would have to kind of focus, right? Like we could see just part of this cell at any one time. Whenever we look through a microscope, we are effectively looking at a cross-section of something. All right, so surround cells, they are semi-permeable. Write this down, and if you know what it means, that's great. If you don't worry about it, because we're going to talk about it shortly. They are made from a structure called a phospholipid. And if you know what that is, fantastic. If you don't worry, because we're going to talk about it shortly. And I just want to make sure you have it written down so that you have that information, and then when we talk about it later, you'll have it in a couple spots. They contain things. We're not going to go into a ton of detail about these things right now, but they're actually really important. Without the things that are in a cell membrane, the cell would be unable to move really critical substances in and out. This right here, this purple wrinkly shell, is like a cell membrane. Can't really see the things that are embedded in it. These little blobs with like a pink circle and like two like squiggly lines are drawings. That's not really what they look like. It's not too far off. These are what phospholipids look like, and again, we'll talk more about those in just a second. These other things are the things that are embedded in the cell. This is one, this is one, this is one. These are things that are stuck to the outside. Sometimes they're stuck to other things that are embedded in the membrane. These structures generally facilitate the transportation of materials in or out of the cell, and it's really critical. I'm not going to talk about the details of them. There's lots of different kinds. They do lots of different things, but we are going to talk in general about how they work to move things in and out of a cell. Cells are made from two separate layers, and we're not going to look at this in a ton of detail, but it turns out that from a physics and chemistry standpoint, the reason that cell membranes work is because of this. They're made from two layers of the exact same thing, one facing one way, one facing the other way. It's almost like two pieces of Velcro that are stuck together. You can see it here most clearly. This is one layer, this is another layer. In this diagram, they've been peeled apart so you can see, but in a real cell, they're sort of stuck together like this. One part of the phospholipid is on the outside of the outer layer and the inside of the inner layer. And then in the middle are these squiggly things, and we'll talk about all of those things. It is the physical and chemical properties of phospholipids that determine what can get in and out of a cell. And it is the physical and chemical property of a phospholipid bilayer that makes it such an effective packaging material for biological organisms. And then the last thing is that when we talk about cell membranes, we are specifically referring to the membrane around the outside of a cell. That's what the cell membrane is. When we talk about membranes in general, there's lots of things in organisms that have membranes, and it turns out that much of the stuff that we're talking about with cell membranes also applies to the membranes that surround other structures, organelles, inside the cell. So for example, the mitochondrion has a membrane. The endoplasmic reticulum is membrane-based. This is an animal cell. We're not going to see any chloroplasts. They have membranes. The nucleus has a membrane. The Golgi apparatus, which is right here, has a membrane. So these, and lysosomes have membranes, ribosomes don't, interestingly. But a lot of the things that you were looking at, vacuoles, lysosomes, in cells have membranes. It's like little cells within a cell. They're not real cells, but they're similar to cells, and the membranes are similar. We're not gonna deal with this too much, but it's worth kind of picturing. One of the really cool things about how cells do their jobs is that if you have packages within packages, it leads to a really easy way to bring things in and to export things out of a cell by kind of like joining the packaging together. And you'll see examples of that next week or the week after. I want to talk about two things in a little bit more detail. The first are these basic pieces of a cell membrane, which are called phospholipids. And phospholipids are categorized. Remember how we had 4 biochemical types of biochemical compounds? There were proteins, there were carbohydrates, there were nucleic acids, and then there's lipids. These are generally categorized as a lipid, but they're sort of a special lipid because they actually have other stuff. And what makes phospholipids so special is that part of them is polar and part of them is not. And we are not going to recap polarity right now in detail. But remember when we were talking about water, and I showed you how water molecules have 1 oxygen atom and 2 hydrogen atoms, and because of the chemical nature of these atoms and like physical properties of how close the electrons are to the protons and all sorts of stuff, it turns out that that part of water molecules has a little bit of a negative charge, and part of water molecules have a little bit of a positive charge, like a magnet. Well, phospholipids work the same way, except they have a part that has a little bit of a positive charge, and a part that has a little bit of a negative charge, and a part that doesn't really have any charge at all. So what we have here is a little bit of a negative charge on one end and a neutral charge at the other end. And what that means is that they have kind of a mixed polarity. One part of them acts like water. This part kind of acts like water. It sticks to things that have electric charges, like water. One part of it does not have a charge, and it doesn't act like water. This is kind of like oil. Oil and water normally don't mix, but in a phospholipid, they're physically attached to each other. And that ends up being really important, and again, we'll look at that in a lot more detail when we start talking about cell transport mechanisms and how they work. The phosphate group is this part up here. This is called a phosphate group, and it's right here in this diagram of a phospholipid. You don't necessarily need to know the details of all this stuff, but you'll hear it and you'll read about it. The phosphate group is polar. And you can kind of remember that because it has a P, right? So phosphate, P for phosphate, P for polar. Okay? And then it also has a little piece of something that's actually a sugar. It's a special kind of a sugar called a sugar alcohol. It's called glycerol. And that's right here. And the function of this in phospholipids is it connects the polar part, the phosphate group, to the non-polar part, which are lipids. They're a special kind of lipid called a fatty acid. And that's just a weird name, but actually they're just like, when you guys were doing your pattern matching activity, those really long things that you were looking at, those are fatty acids. Right? And it basically is just a long chain of carbon with a bunch of hydrogen and a little bit of oxygen. These are non-polar. They don't have any sort of overall electrical charge or partial electrical charge. They won't stick to water. In fact, the water will stick to itself, and these will kind of push the water out. The water like grabs onto itself and sort of pushes these out, and so they repel. Actually, there's a repulsive force from these non-polar fatty acids on water. The polar part of a phospholipid sticks to water. So if you think about our cells and the way that they are designed, they've got two layers. They've got a phospholipid bilayer, so we've got, say, an outer layer and an inner layer. And on the outside, the polar part of these cells likes to stick to water. So we're going to have lots of water out here. On the inside, the polar part of the phospholipids will like to stick to water. So we're going to have lots of water on the inside of a cell. And in between, the non-polar part, these fatty acids, don't like to stick to water. And so because of that, the water kind of, it partitions it. This is what causes the partitioning to begin with of cells. And it seems like a pretty elegant engineering design, and a lot of people are like, well, that's crazy, how did that ever happen? And it turns out, it happens on its own. We may or may not have time in this class to do this. It's a really quick little experiment, but we can make basically like the same principle behind soap bubbles is what governs the self-assembly of phospholipids to make membranes. If you take phospholipids and put them in a container and shake them up, they will make stable bubbles on their own. They will form these membranes on their own. So one of the really cool things about all of this is that it's not something that has to be like really meticulously engineered. Have you ever put together one of those three-dimensional puzzles where you have like, it's not just flat, you're like building a globe with puzzle pieces? And there's that point where you're like, you have to use your one hand to hold the puzzle, and then you have one hand that's stabilizing the part that you're trying to attach. And then one hand is holding a piece. One hand is holding like the connector piece on this side, and one hand is holding the connector piece on this side. So you've got all five of your hands in on building this puzzle, and it just doesn't quite make sense because that's more hands than most of us have. But when you look at something like this, it doesn't take anything, it just happens on its own. These things naturally stick together, they naturally form bilayers, and those bilayers naturally partition things into an inside and an outside. So it's a really critical part of biological organisms, and the emergence of phospholipid bilayers was a really critical part in the emergence of life on this planet. And then the last thing to think about, and it's this, we'll see some examples of this. We can watch it happen, but only with computer models. One thing that's cool about this is, since all of these little phospholipids are their own individual piece, and since they're not stuck together with like really strong molecular bonds, these things are all stuck together because of polarity, the same way that water sticks together. It turns out that a cell membrane is not like a balloon or a plastic bag. It's fluid. The whole thing kind of flows a little bit. And not just in this sense, right? Not just in the sense that we can like push on a cell membrane the same way that we can push on a balloon, but in the sense that if you were to look at any one particular cell, you might find this structure here at one point, and then it might actually kind of move over somewhere else. Things can be pushed into or pulled out of the cell membrane because of its fluid nature. It's kind of like if you covered a swimming pool with ping pong balls, right? It would be covered, but also you could like dive in and you'd be in the pool. You could go down below them. You could come up above them. You could put a little rubber raft in there. That's how a cell membrane works. It's not that fluid. They stick together a little bit more than that, but it's not like a balloon or a plastic bag where like the membrane is fixed in place. And then the last bit of information has to do with this idea of semi-permeability. And semi-permeability is actually really common. Most things are semi-permeable to a certain degree. We use containers all the time. It's kind of our thing. But when you think about the containers you use different ones for different purposes. If, for example, you wanted to carry something home from school, you might put it into a paper bag, right? Makes sense, I put things in a paper bag. But what if you had some soup, some leftover soup? Would you fill up your paper bag with soup and then carry it home? Like, is this something that any of you would feel pretty good about putting into your backpack, taking home? This is really important water. Your grades depend on this. I need you to take this water home, okay? So, you on this? Can you take this? Just put it in your backpack. It's in a container, so you're fine, right? This is a terrible container for water. But if you're carrying dry stuff, it's fine. You can put I could put all this stuff on my desk in there. And virtually all materials work like that. Basically what it means is they hold some things and they don't hold other things. And when it comes to, well, I don't know where my pen is. Let's use this one. When it comes to cell membranes, they have some real specific things that they can keep in and out, or that they let pass through. So there are some things that can move through a cell membrane like it's not even there. And these things, in general, fall into a couple of different categories. One is they're small. So things that are really, really small have a pretty easy time getting through a cell membrane. The other is that things that have no electrical charge overall, or partial charges, so non-polar things are easier too. Small is because in order to get through the cell membrane, it has to kind of squeeze in between these phosphate groups on the phospholipids. If they're too big, they just can't fit. Physically, they get stuck. Non-polar is easier because if they're polar, they'll stick to the phospholipid heads. So something polar, something that's really charged, will hit the polar side of the phospholipid membrane and get stuck. It doesn't move through. And that happens on either side of the cell membrane, so things can get trapped inside or outside of the cell, depending on how big they are and what their charge is. Some examples of things that can move through a cell membrane: oxygen gas can move through a cell membrane pretty easily. Carbon dioxide can as well. The rest of these things you don't have to write down. It's not super important. The one thing that I want to point out is that while water is polar, but it's also small. So it turns out water can move through a cell membrane, but it doesn't really fast. It goes really, really, really slow, in fact. And there are special structures in cell membranes that allow water to move in and out, called aquaporins. But cells on their own are pretty watertight. They're not 100% watertight, but they're pretty watertight. And then lipids and hydrocarbons can move through as well, and we're not even going to be concerned about those, so you don't have to write that down. There are other things that cannot move on their own through a cell membrane, and those are things that are either large or have polarity or are charged, or both. So larger or polar molecules have a tough time getting through that cell membrane on its own. There are some things that are really small, but they have a charge. Things like hydrogen ions. This is, those of you who know a little bit of chemistry, a hydrogen ion is a proton, one single proton. And this right here, Na, is sodium. Sodium ions cannot move in or out of cell membrane. And it's a really good thing that they can't, because as it turns out, the fact that these things can't go through is what allows life to exist on this planet. We're not going to talk about this stuff at all. If you take AP biology, you will. But it turns out that one of the ways in which cells can make things happen is by pumping hydrogen ions or sodium ions in, and then letting them trickle back through a special kind of protein that does something. It's like using water behind a dam to run a generator. That's effectively how it works. These proton pumps and sodium pumps make your cells do all sorts of things. If they didn't exist, our cells would be pretty worthless. And then there's other larger molecules like glucose and most other carbohydrates, large carbohydrates and proteins. They can't get in and out of cells on their own. They need help to do that. We'll look at that stuff next week or the week after. All this stuff is really having to do with the idea of transport mechanisms. But what I want to talk about for the rest of the class, what you're going to be looking into is this idea of semi-permeability. And so what we're going to do is we're not going to work with real cell membranes. Not because they're dangerous, because they're kind of difficult to work with. But what we can do is we can work with a different kind of membrane. This is not a cell membrane. It doesn't work exactly like a cell membrane does. But like a cell membrane, it's semi-polar. And some of you might have used something like this before. This is called dialysis tubing. Has anybody ever used dialysis tubing for anything? It's really fun. Does this look like a tube to you, by the way, Caroline? Is this a tube? No, it's not. It's just a sheet of plastic. But this is, you know, when it comes to this kind of stuff, I'm a total goofball. But as it turns out, this is a tube. And here's how you can see that. If you take this and you put it in a container with water, and you let it soak for just a minute or two, and we're not gonna have to let this soak for long, then we'll see that it's a tube. And dialysis tubing is not the same as a cell membrane. Cell membranes let very small, non-polar things in and out, and they generally block larger or polar molecules and charged substances. This dialysis tubing is also semi-permeable, but it's permeable to different things. And so what that means is we don't know what this lets in and what it lets out. We have to find out, and that's going to be your job today. Okay, I think this is probably good to go. So this is, I know this is just like the dorkiest thing, but I love opening these things up. So once you've soaked it for a little bit, if you take your fingers and you kind of push and squeeze it side to side like this, eventually you get it to like start like opening up, and it's just so satisfying to turn this into a tube. Might need just a couple more minutes. Let it soak for a little bit more. Oh, it's just so satisfying. So what you're going to do is you're going to answer a few questions about what can move in and out of dialysis tubing. In order to do this, you're going to have to design an experiment to test it. So here's the three things that you're going to look at. And I don't know how far you'll get today. You may get them all. You may just get one or two, and that's fine. But the things that we want to investigate is something called starch, which is a carbohydrate, something called iodine, which is a small molecule. Iodine is an element on the periodic table. Salt, which is made up of two pieces that come apart in water. They're both charged, sodium and chloride ions, and then water itself. So what we're trying to figure out, I keep putting my pens down and I don't know where they are. What we're trying to figure out is if this is a piece of dialysis tubing, can water get through that? That's one of the things we're looking at. We're looking at sodium or chloride ions. Can those get through? We're looking at starch. And we're looking at, this really should work more fluidly, and we're looking at iodine, okay? So these are the things that we're trying to test. Let's see if my dialysis tube is ready to be opened yet. I'm feeling confident. Yeah, I can feel it. It's got the wrinkle. The wrinkle has started. So once you get the wrinkle started, and you kind of peel it apart a little bit, then if you put it under a little tiny dribble of water, then it just sort of fills, it'll like fill up and sort of, the water will sort of start flowing through on its own. So now we can see that this is indeed a tube and not just one piece of plastic. And the way that you're gonna work with these, it takes, so there's a little bit of dexterity that goes along with it, because you need to be able to tie knots. It's the kind of thing where sometimes it helps with a couple of people, but basically what you're going to do is you'll take a string and you will tie it with it. I mean, it's not my fault that my desk is this messy. Other people have created this. You take one end of it and you kind of twist it like this, and you can tie a string around that end. And this is much easier with a couple of people, but it's not impossible to do on your own. And then you can fill up the tube. And you can fill up the tube with water, or you could fill it up with like water mixed with sodium and chloride ions by putting salt in there. You could fill it up with something that has starch or iodine in it as well. And then you can tie another piece of string to the top, and then you have a little container, and you can see that it's like at least reasonably watertight, right? So if I have this little blob of water in this dialysis tubing, what I want to know is, can more water get in, or could water come out of here? I want to know, can salt come in or out? I want to know, can starch go in or out? I want to know, can iodine go in or out? And figuring all of these things out is a little tricky. I'm gonna leave, water is gonna be the last one. We'll deal with that last. Some of you may or may not get there at all, but it's kind of a fun one to look at. The other two, though, I'm gonna tell you how we can make the measurements. With salt, we have a way of detecting salt, and it's not by tasting it. I mean, of course, that would work, but we're not gonna do that. Instead, we're gonna use this digital multimeter, which can allow us to check resistance. And basically what happens is, when water has salt in it, its resistance drops. So we can check to see whether or not something has salt in it by looking at its resistance. So this plain water right here has a resistance, you probably can't see that, but it's around 14,000. 14,000, what's the unit of resistance? Does anybody know? Anybody taking, it's horseshoes. It's got about, now it's gone up a little bit, it's about 16,000 horseshoes of resistance. The real unit for horseshoes is ohms. Have you ever heard that? Yeah, kiloohms. So when we have pure water, it's about 16,000 16,000 ohms. But if we add water to that, salt to that, then that resistance drops a lot. So it was 16, right? Now it's 10, 1. Now it's dropped by a factor of more than 10 when I added the salt. So we just look for lower resistance. Now it's down below 1. It's like 0.9, 0.8. It's about half, about a 20th of what it was before. So it's really dropped a lot. And so that's a way that we can test for salt. And starch is a little interesting too. So starch we can see. So I have some starch here, or maybe I have some starch here. I actually can't remember. One of these has starch in it, and one of them doesn't. They both look the same, right? One of them has baking soda. It's not the same as starch. But as it turns out, if you have starch in something, then when you add a little bit of this iodine, so iodine is kind of this brownish color, really dark brown. If this is starch, when I mix the iodine in, it should turn purple. Does that look purple? I think that just looks brown still. All right, so hopefully this is the starch. Guess we'll find out. If this is the starch, this should turn purple. Oh yeah, you can see that, huh? So we can detect the presence of starch by mixing in some iodine. If it's got starch in it, the whole thing turns purple. If it doesn't have starch in it, we don't see anything. Here is your task, and it's not super easy. You need to tell me how you're going to use these materials, the conductivity meter, the multimeter. We've got iodine, we've got starch, we've got salt, we've got water, we've got beakers, we've got string, we've got dialysis tubing. And your job is to tell me how you're going to figure out which of these things can move through the dialysis tubing or not. Remember, dialysis tubing is not a cell membrane. They're two different things, but like a cell membrane, dialysis tubing is semi-permeable. Some of these things can cross the membrane, and some of them can't. So you can work, I think groups of 3 would probably work best for this, because there's a lot of stuff to test, and potentially a lot of like dialysis tube setup. And honestly, just tying the knots is difficult enough. So find a person or 2 to work with. If you want to take a break before you get started, that's fine, but be back here at 2:30. I think so, yeah. But I want you to be back here at 2:30, and then before you get anything, I want to see your plan. How are you going to determine whether these things can move through dialysis tubing?